So, hello, good evening, students. Myself, Ankit Malik, PhD scholar and a PMRF fellow from IIT BHP from the Department of Pharmaceutical Engineering Technology. And the topic which we will cover today for the basics of amino acids and proteins are introduction to amino acids, introduction to proteins, and introduction to proteomics. And the course instructor for the same is Professor Sanjeeva Srivastava from IIT Bombay. So let's start from the topic introduction to amino acids. So this is a week one session. So we'll just cover the basic things today, like some basic, entirely basic things. What are amino acids? What protein stands for? What are proteomics and what are their works and what are their techniques? Okay, we'll not go into that deep knowledge. Okay, so let's start. So what are amino acids? So all you know that the building amino acids are the building blocks of proteins. So amino acids are organic compounds that serve as the building blocks of proteins. So protein in turn are essential macromolecules in all living organisms, playing roles, structure, function, and regulation of tissues and organs. So there are totally 20 standard amino acids, which with a unique side chain or R group that gives it distinct properties. So in this figure, as you can see, the the alpha amino acids because of the alpha carboxylic and alpha amino groups, which are this one, this is all C alpha, okay, this is alpha carboxylic and the alpha amino groups. So here pKa1 is the alpha carboxylic group and pKa2 is the alpha ammonium ion. Okay, so in the physiological range, pH range, so both carboxylic and amino groups are completely ionized, as you can see in the structure in the figure also. So basic structure of amino acids. So each amino acid has a central carbon atom known as the alpha carbon, as shown in this diagram. Okay, so carbon bonded to four groups, that is amino group NH2, carboxylic group COH, hydrogen atom, that is the H, and R group, that is side chain. So amino group, so this group can accept a proton giving it basic properties, and carboxylic group can donate a proton giving its acidic property to any amino acid. Okay, so then hydrogen atom, which is attached to the alpha carbon. Wait, I'll show you once again. Okay, then the R group, there is a side chain. So this is a variable group that determines the identity and properties of the amino acid. It can range from a simple hydrogen, which is a glycine, to a more complex ring structure. Example is in tryptophan. Okay, so let's decode amino acids. So you must know this is a diagram, this is a structure of tyrosine. So only tyrosine can be named by the four things, by their names, by the structure, by the three-letter code, or by their one-letter code. So as you can see the structure of tyrosine here. So we we can identify this amino acid by their structure, then by its name tyrosine, then by its three-letter code, that is TYR, and only one letter code, that is a Y. Okay. So then the classification of amino acids. So amino acids can be classified based on the properties of their R groups. So the non-polar amino acids, which is hydrophobic in nature, then the polar amino acids, that is hydrophilic in nature, then charged amino acids. So we'll start from the non-polar ones. So this non-polar hydrophobic amino acids have side chains that do not interact well with water. Examples include valine and leucine. Then on the other hand, polar hydrophilic amino acids, so these have side chains that can form hydrogen bonds with water, making them soluble. Example serine and threonine. Okay, and the last is charged amino acids. These are further divided into acidic, is a negatively charged, example is aspartic acid, and basic positively charged, example is lysine amino acids. Okay, then there are two types of amino acids which we characterize usually. There is essential amino acids and non-essential amino acids. So essential amino acids cannot be synthesized by the human body and must be obtained from the diet. So there are 9 essential amino acids including lysine, methionine and tryptophan. On the other hand, non-essential amino acids. So the body can synthesize these also. So they cannot required in the diet. So examples include alanine and asparagine. So we'll cover the properties of amino acids now. Amino acids exhibit several key properties. So the first property is zwitterionic nature. So at a physiological pH, amino acids exist as zwitterions, which means both the amino acids can act as a positively charged amino group and a negatively charged amino group carboxylic acid at the same time, simultaneously. So this is the first nature of the property of amino acids, that is zwitterion nature. So the next property is isoelectric point, in which we known as a pi. So this is the pH at which the amino acid carries no electric charge in it. So it varies depending on the amino acid groups. Amino acids R group, that is a side chain or side groups. Okay. So the last property is optical activity. So except for glycine, all amino acids have chiral centers and can exist in L or and D forms. So in protein, only L amino acids are used. Okay. So now we'll understand the peptide bonds and their protein structure. So amino acids link together via peptide bonds. I'll show you in the structure as well. So which are formed in the condensation reaction between the amino group of one amino acid and the carboxyl group of the another. So this bond formation releases a molecule of water. So the sequence of amino acids in a protein determines its primary structure. So which folds into complex 3D shapes to form secondary, tertiary and quaternary structures. So these structures are critical for the protein functions. So I'll show you the structure now. So the amino acids can form a peptide bonds as I already told you. So the residue peptide units, dipeptides, tripeptides, oligopeptides and polypeptides. So usually proteins are the molecules consist of one or more polypeptide chains, as you can see in the structure. Okay. So in this structure there are totally 20 different naturally occurring amino acids are there. So now there is a chart for covalent structures and their abbreviation for the standard amino acid proteins. So here in this table, so the amino acids with non-polar side chain and their examples are there with their structures, with their residue mass, with their average occurrence in proteins, their pKa carboxylic value, their pK2 ammonium values, and their pK side chain, that is R group if something is there. Okay. So amino acids with non-polar side chains are there. Amino acids with uncharged polar side chains examples are there. So amino acids with charged polar side chains. I'll dictate one by one the examples. So amino acids with non-polar side chains are glycine, alanine, valine, leucine, isoleucine, methionine, proline, threonine, tryptophan. And the amino acids with uncharged polar side chains example are serine, threonine, asparagine, glutamine, tyrosine, and cysteine. And the amino acids with charged polar side chains. Okay, so lysine, arginine, histidine, aspartic acid, and glutamic acid. So cover all these things and with their values. So next we'll cover the biological functions of amino acids. So basically amino acids are not only the building blocks of protein but also serve other vital functions like metabolism and gene expression. So there are the amino acids involved in various metabolic pathways including the synthesis of neurotransmitter and their hormones. And the gene expression, certain amino acids play a role in regulating gene expression and cell signaling. Mostly with the polar nature, amino acids plays the role of gene expression. Then there are applications in biotechnology and medicine. So amino acids have significant applications in biotechnology and medicines, are recombinant protein productions, their therapeutic use, like some amino acids are used in treatments of certain diseases such as amino acid infusions in patients with their metabolic disorders. So now we'll look into the introduction of proteins, what proteins are. Yes. So proteins, everybody know proteins, know about the in bodybuilding or some supplements, through supplements, everybody knows the name proteins. So proteins are naturally occurring polymers made up of amino acids and linked together by peptide bonds. Okay. So mostly my proteins are macromolecules obtained from one or more amino acid chains linked by peptide bonds. So they are natural polymers of amino acids. So it contains nitrogen, carbon, hydrogen, and oxygen. So they act as a biological catalyst from structural parts of different organisms participate in different cell reactions. Okay. So now we'll these are some basic things. So now we'll the, the classification of proteins. So basically they are divided into three classes. So simple, conjugated, and derived. Okay. So now we'll cover one by one their definitions, their examples. So there are three types of proteins. Very simple. In simple, it is globular proteins and scleroproteins. So mostly in globular proteins, the examples are albumins, globulins, prolamins, glutenins, histones, protamines, globulins. And in scleroproteins, it covers collagens, elastins, and keratins. Whereas in the conjugated one, the examples are nucleoproteins, chromoproteins, glycoprotein, phosphoprotein, lipoprotein, and metalloprotein. And in the last category, which is further divided into two subclasses, that is primary derived proteins and secondary derived proteins. So the examples are coagulated proteins, proteins, and metaproteins. And in the secondary ones, proteases, peptones, polypeptides, simple peptides, and amino acids. Okay, so now we'll cover each and every example which covered in the classification. So first, simple proteins. So their simple proteins are composed of only amino acid residue. So the first thing was globular proteins. So these are spherical or oval in shape and mostly soluble in water or other solvents. So one more thing. So this way we have covered the classification of proteins according to their nature, and one will cover according to their structures. Structures of proteins, the composition and function of proteins. Okay. So this is according to their function of proteins. In the next phase, we'll cover the classification of proteins according to their structures. Okay. So in globular proteins, so the first thing is albumin and globulins. So these are the globular proteins found in cells like serum albumin, oval albumins, there is eggs and lactalbumins, and secondary glutenins, prolamins, histones, globulins, and protamines. So and lectins as well. So we'll cover the protamines and lectins, which are the important ones. So and protamines, which are strongly basic and resembles histones, but are smaller in size and soluble in the ammonium things. And protamines are also found in association with the nucleic acid and various form proteins. And the last is lectins, which are carbohydrate binding proteins and are involved in the interaction between cells and proteins. So they help to maintain tissue and organ structures. Okay. Hmm. So and the next it covers fibrous proteins. So it covers these are fiber-like in shape and insoluble in water, and the examples are collagens, which are connective tissue proteins. On the boiling with water, it becomes soluble and digestible. And the other is elastin. These proteins are found in elastic tissues such as tendons and arteries. And the last is keratins. So mostly I've heard this keratin when it comes to hair. So mostly people say the whole hair fall is because of keratins. It is a hair protein. So that is the thing the keratin is. So keratin is found in the protein of skin, protein, hairs and nails. Then it comes to conjugated proteins. So besides the amino acids, so these proteins contain a non-protein moiety known as the prosthetic group or a conjugating group. It covers nucleoproteins, chromoproteins, phosphoproteins, glycoproteins, lipoproteins, and metalloproteins. Metalloproteins, it contains some various metals which are attached to simple proteins. Then lipoproteins, which is the combination of proteins with lipids found in the brain and membrane. Then glycoprotein, which is consists of amino sugars like carbohydrate and amino acids or sulfate and sugar acids also. And the phosphoproteins. So along with the amino acid contains a phosphate group as well. The R1 is the phosphate group. So example is milk casein. And the next is chromoproteins. So which are soluble in proteins combined with chromophoric is a colored group. So the colored group is heme and riboflavin. And okay, so this is done. So now we'll cover the derived proteins. So the derived proteins are the denatured or degraded products of simple and conjugated proteins. So the classes are further subdivided into different groups. So different groups are primary derived proteins, secondary derived proteins. So in primary derived proteins, so it is coagulated proteins. So these are the denatured proteins produced by heat, acids, or alkalis. For example is albumin. It is a white egg white. So in coagulated proteins, so the next is proteins. So these are the first or the earliest product of protein hydrolysis by enzymes. So the next is metaproteins. So these are second product of protein hydrolysis. So and the stronger acids are used. Example are explained alkali metaproteins. And the second category for the primary derived proteins is the secondary one. So which are degraded due to the breakdown of peptide bonds, product of proteins. So now we'll cover the classification of proteins based on the structures. Hmm. So here we have a primary structure, a secondary structure, a tertiary structure, and quaternary structure. So totally there are four levels of protein classification according to their structure. So mostly, as you can see in the primary protein structure, is a sequence of chain or sequence of chain of amino acids. So it gives the detail about the amino acid sequence of a protein. So it tells about the number of amino acid residues in the protein and also about the sequence of amino acids. So how many amino acids are there? So which it can be clearly visible. So it comes in primary protein structures. So it is stabilized by peptide bonds. So each component of an amino acid is called residue or a moiety in this in primary protein structure. So it starts from the amino terminal, which is called as the N, and ends in the carboxylic terminal. The next one is secondary protein structure. So as you can see, there is the alpha helix and petite sheet is there. So hydrogen bonding of the peptide backbone causes the amino acids to fold into a repeating pattern. Okay, so it formed by hydrogen bond basically between the backbones of the atoms. So three most important secondary structures in protein are in secondary protein structures are alpha helix, then B plates as you can see in the structure, and the beta turns. So, well, this is one alpha helix. I'll tell you about this. So, alpha helix is the most common secondary structure which repeat every for I think 5.4 units. So, it's the simplest arrangement of a polypeptide chain which are proposed by one scientist, Pulling and Corey. So, the alpha helix one is the most common because it makes optimal use of internal hydrogen bonds. So the interaction between the amino acid chains can stabilize or destabilize the alpha helix things. Okay. So the next is tertiary protein structure. So as you can see, there is a 3D folding pattern of a protein due to side chain interaction. So as you can see here, the tertiary structure will have a single polypeptide backbone consisting of one or more secondary structures. So it can be defined by atomic coordinates and it is stabilized with the help of both the covalent and non-covalent bonding. And the last classification is according to structure is the quaternary protein structure. So in this we call the protein consisting of more than one amino acid chain. So in quaternary, the protein which have more than one polypeptide subunit, which do not have a permanent, which is a covalent interaction between the subunits like their disulfide bonds, so which are classified under quaternary structures. So bond stabilizing quaternary structures include hydrogen bonds or hydrophilic interaction, hydrophobic interactions, van der Waal interactions, and the function of this all the proteins which we'll cover in the next slides. So this I've covered all the things. So now the properties of protein. So it covers solubility, molecular weight, shape, Zwitter already discussed in the previous slides. Then precipitation and coagulation. So the solubility. So proteins have a huge sizes and therefore form a colloidal solution in water. Then a molecular weight. So the molecular weight of proteins varies depending on the number of amino acids. They have higher molecular weight. So the higher molecular weight ranges from 40 to 4000. And the shape varies. Globular shape. Insulin has a globular shape, and albumin has an oval shape, whereas fibrinogen has an elongated shape. Then Zwitter, which I've already covered. Then precipitation. So proteins get hydrated when salts such as ammonium sulfate are added. Dehydration also results from the addition of salts of heavy metals like lead and zinc, etc. And alcohols also dehydrates protein. So tannic acids and picric acids also dehydrate protein. And the last one is coagulation. So it is a semi-solid or solid precipitate of protein. It is irreversible. Example is albumin. So now we'll look into the proteomics. What proteomics are? So we'll take a break for 2 minutes. We'll just continue. Hanji. So the introduction to proteomics. And so what is proteomics? So basically the study of entire set of proteins is a proteome expressed by a genome, cell, tissue or any organism. So unlike the genome, the proteome varies with time, environment and cellular state. So it's a large scale study of proteins, particularly their structures and functions are explored one by one. So it's a branch of molecular biology concerned with the comprehensive analysis of proteins in a particular organisms or any tissue. Okay, so the term the proteome refers to the entire set of proteins expressed by a genome or any tissue. So unlike the relatively static genome, the proteome varies from cell to cell and from tissue from time to time, reflecting the organism's dynamic responses to different conditions. Okay, so what are the importance of proteomics? So functional understanding, uh, where the proteins are essential to all the cellular processes. So proteins are the workhorses of the cells, as you know. So which involved in virtually all cellular processes. So understanding the proteome provides insights into cellular mechanisms, pathways, and their functions. And the second importance is disease diagnosis. So it identifies biomarkers for diseases like cancer. So many diseases, including cancer as well, or any injury also, and some asthmatic things, Alzheimer's. So are linked to abnormal protein expressions. So proteomics can identify diseases biomarkers, leading to earlier and more accurate diagnosis, and can also aid in the development of a targeted therapies. And the next is drug discovery. So proteomics plays a crucial role in drug discovery and development by identifying potential drug targets and understanding drug protein interactions. And the last importance of this proteomics, system biology. So proteomics is a key component of system biology which integrates data and from genomics, transcriptomics, metabolomics and other fields to understand the complexity of biological systems. So next is types of proteomics: structural, functional, and special. We will cover one by one now. So in the structural proteomics, so the ultimate aim of the structural proteomics is to build a body of structural information that will help predict the probable structure and potential function for almost any protein from knowledge of its coding sequence. Okay, so the next is functional proteomics. So it refers to the use of proteomics technique to analyze the characteristics. So to analyze its characteristics of molecular protein networks involved in a living cells. The last type of proteomics is expression proteomics, which refers to the quantitative study of a protein expression between sample differing from some variables. Okay, so the next is a techniques in proteomics. So we'll cover one by one. I'll tell you their system, their importance, their use. So the first one is mass spectroscopy. Second is 2D gel electrophoresis, liquid chromatography, protein microarrays, and bioinformatics. Okay, so they all employs a variety of experimental techniques and analytical methods. So the first is mass spectroscopy. This is the most common technique used in proteomics. So it identifies and quantifies proteins by measuring the mass to charge ratio, okay, of the ionized particles in the proteins or any amino acids. So it can analyze complex protein mixtures, determine post-translational modifications, and provide information on protein structures. And the second technique is 2D gel electrophoresis. So 2D gel electrophoresis separates proteins based on their isoelectric point and molecular weight. It is useful for visualizing the protein composition of a sample and comparing protein expression under different conditions. And the third one is the liquid chromatography. So often coupled with LCMS, mostly with mass spectroscopy and liquid chromatography, so which is called LCMS. So this technique separates proteins or peptides in a mixture. It enhances the sensitivity and resolution of mass spectroscopy, so allowing for more precise protein identification. And the fourth one is protein microarrays. So these are high throughput platforms that can detect and quantify protein interactions, modifications, and their functions. So they are used in functional studies and biomarker discovery. And the last is bioinformatics. So for the large data sets generated in proteomics require sophisticated bioinformatic tools for data analysis and their interpretations. So these tools help in protein in identification, quantification, and functional annotations, and pathway analysis as well. Okay, so there is a diagram which determines the understanding of omics, like genome, transcriptome, proteome, and metabolome. So I'll explain one by one for the use for the genome, transcriptome, proteome, and metabolome. So the genome is a complete set of DNA within an organism, including all of its genes. So it contains the instructions needed to build and maintain the organism encoded in the sequence of four nucleotide bases. So the four nucleotide bases are adenine, thymine, cytosine, and guanine. So the application for this genome here is the disease research in genetic disorder or in cancer, evolutionary biology, and personalized medicines. Okay, and the next is transcriptome. So this the transcriptome is the complete set of RNA transcripts produced by the genome. As you can see, it comes from the genome, so at any one time. So this includes mRNA, tRNA, rRNA, and non-coding RNA. Okay, and it has dynamic nature, gene expression, and transcriptomics. And the application for the transcriptome RNA is to understanding gene regulations, identifying diseases biomarkers, and studying responses to some environmental changes. Then it comes to our proteome. Okay, so I've already told you the definition and their uses and their key points and applications. So I'll cover once again. So the proteome. So proteome is the entire set of protein expressed via genome cells, tissue, or any organisms at a certain point of time. So proteins are the functional products of genes and are essentially to virtual all biological processes. And the application of proteome is in drug discovery, in biomarker identification, understanding cellular processes and signal pathways. And the last one is metabolome. So metabolome is the complete set of small molecule metabolites found within a biological sample, such as a cell, tissue, or any organisms. So these metabolites are the intermediates and product of a metabolic reaction. Okay, so the key points for these metabolome is its metabolic activity, its metabolomics and varied components such as hormones, lipid, sugars, amino acids. Okay, and the application of this metabolome is in the understanding of metabolism and metabolic disorders, then nutritional studies, and drug metabolism and their pharmacokinetic studies. Okay, so these are some challenges in the proteomics. So in this it has some complexity, dynamic range, and the low abundance proteins. So as you can see, as we go through this genome to proteome, the complexity is increased. Okay, so here the in complexity, proteomics faces several challenges, including like the protein mixture complexity, the dynamic range of protein concentrations, and the detection of low abundance proteins. Despite these challenges, advances in technology and methods continue to enhance our ability to study the proteome. So we'll cover one more thing is future aspects. So the future of proteomics lies in integrating data from other omic fields, other from like genome, metabolome, and proteome. So developing more sensitive and accurate analytical techniques and improving bioinformatic tools. So these advancements will further our understanding of biological systems and contribute to some personalized medicines where treatment can be tailored to an individual's proteomic profiles. Okay, so this is all for the day. So in the next topic, in the next week, we'll cover these topics into some deep manners. So today's session was of all basic briefs, what the terms are. Okay, so now you guys can leave. Okay, thanks for the day.